So welcome everyone to the third and the last session of the first Soapbox Science St. John's event. My name is Ozgen. I am one of the three organizers and your host today. I also want to mention Sarah and Christina, the other two Soapbox Science St. John's organizers. They're also present here, making sure that everything works smoothly in the background during the event. And I also want to thank them. They have also done a lot of work for more than 6 months now and to get this event together. So in terms of housekeeping, I also want to mention a couple functions of Zoom. So as you noticed that you were muted, your camera was off when you joined. Please remain muted with your camera off because this is for your privacy. As you notice, we are recording this session to share with other groups that they couldn't join us today. But doesn't mean that we want you to be silent in the background. We encourage you to engage with our speakers using the chat function to ask questions. Christina and Sarah will be monitoring the chat. You can send your questions to the chat box, and they will keep a list of questions, and I will be asking the questions in the second part of our session, in the panel session. You can also use the button functions of Zoom during the presentations of the speakers, such as clapping, yes or no, to engage with the speakers simultaneously if the speakers will asking you questions. And also, before we talk about science, as an immigrant student myself, who lives, works, and studies in Newfoundland, and as many of you here today, as settlers, I would like to acknowledge and honor the original occupants of these unceded lands of the Mi'kmaq and historically of Beothuk. In Labrador, settlers live and work on the traditional lands of the Inuit of Nassavut and Nunatakavut and the Innu of Nitassinan. So very shortly, I will introduce the speakers to you, and they will each talk about their science, their research, their work for 3 to 5 minutes. We'll then move to panel discussion with 2 prepared questions by myself, Christina, and Sarah. And then I will ask the questions that came from the audience through chat box. And we will do our best to get you an answer in our panel segment. So I'm done with the notices, so let's get started to our session. Soapbox Science was founded in 2011 in the UK to promote women in science and bring science to public, to make science more accessible for public and also encourage women and the science that they do. Typically putting women on soapboxes in public places, but due to COVID-19, this year we decided to move this event to an online format instead of a public engagement. We have wonderful 9 speakers over 3 weeks, and today we are having the last and the third session, and we have 3 very lucky speakers today. We have Julie, Shreyasi, and Kris speaking with us. Julie will be the first speaker today. Julie Turner, she is a postdoctoral fellow in the Wildlife Evolutionary Ecology Lab in Department of Biology at Memorial University. She's broadly interested in animal social behavior, personality, and conservation. And today she will be telling us about hyenas and their social relationships in her talk. Hyenas aren't the enemy. Insight into their lives, personalities, and social behavior. Will be a very interesting speech. And after Julie, Shreyasi Sarkar will be with us. She's a PhD candidate in biochemistry at Memorial University. She's interested in the links between our diet and our intestinal and brain and mental health. She will tell us more in her talk, Missing the Pieces to the Diet-Microbiota-Health Puzzle. And as our last speaker today, Kris Poduska, she is a professor and head of the physics department at MUN. Kris's research looks at the physics of functional materials from traditional ceramic and charcoal to high-tech biomaterials and sensors. And her speech is very interesting, and probably she might surprise you in her talk and make you ask that, wow, you can do that with physics? So I'm all excited to hear all of your speeches today. And we can start with Julie, if you're ready.
Yep. Hi, everyone. I'm Julie, obviously. So how many of you know anything about hyenas, even if it's just from The Lion King? You can use one of those reaction buttons. Yes, Katrine knows anything because she's heard me talk about them. All right, but normally most people don't know anything about hyenas, especially anything other than from what they've heard from The Lion King. And that's just not an accurate portrayal of hyenas. What people do know about hyenas means that they tend to dislike them. They tend to have a bad rap across the world, but hyenas aren't the enemy. A lot of people think they're enemy or they're dirty scavengers or something like that. And really, they're very cool animals. So one of the reasons that we think that people are disliking hyenas is because hyenas are perceived to be scavengers, but like basically all carnivores, they will scavenge occasionally, but hyenas hunt up to 95% of their food. They can bring down a single hyena can bring down a wildebeest all by themselves. So that's something that's many times their size. And sometimes you'll see in nature documentaries, you'll see a lion on a carcass and then a bunch of hyenas on the background, and people don't like hyenas because they think that they're trying to steal the carcass from the lions. But really, it's usually the lion who has stolen the carcass from the hyenas, because hyenas are much more successful hunters than lions are. So hyenas are not the scavengers that they're perceived to be. And not only that, hyenas, the females are very caring mothers. Most of the time, I mean, compared to other species, hyena females spend a lot more time and energy raising their kids than any similar species does. And that's partially because they live in really complex societies that we call clans. And these societies are as complex as those that you see in other primates like baboons, and they're as complex as humans, just in different ways. And you need to, as a hyena, you need to learn a lot of skills growing up in order to fit in that society. So you have to learn a lot of social skills. And that's what I studied for my PhD. I did my research in Kenya in the Masai Mara, which is Maasai land. And here I looked at cubs as they grew up until they reached adulthood. And one of the first things we saw was that males and females develop socially differently, because females stay in the clan that they're born into, whereas males will leave the clan to move to a new clan when they reach adulthood. So females are learning all of the interactions and relationships they need to know within their home, and males, as they get older, start to disengage from their clan and prepare themselves to leave to join another clan. And because we can follow females their entire lives because they stay in that same spot, what we found was that females who have more friends when they're young, they live longer into their adulthood compared to those that don't. And not only that, hyenas that just hang around more individuals tend to have more kids when they're adults as well. So how long you live and how many kids you have is very important for maintaining a population. But what we've also been seeing is that with growing human disturbance in the area, this human disturbance seems to be associated with weakening social bonds within the clan. So one of the things we're looking into in the future, and this is one of the great things of a long-term data set that we have, is seeing how these weakened bonds may potentially be impacting the long-term survival of hyenas. Luckily, they're very flexible animals, but there are still conservation threats to them. And with that, I hope that you have learned that hyenas are really cool animals. I'm happy to answer more questions because clearly there's a lot more to them that I could go over in 3 to 5 minutes. And thanks for listening.
Thank you so much. That was very interesting. And Shreyasi, you can continue.
Thank you for the opportunity, Soapbox Science, and thank you to everybody for being here for this event. So, well, I'm pretty sure you all agree that diet and health have a strong impact in our day-to-day lives. So from maintaining physical well-being to mental well-being as well, diet and health go hand in hand. So, however, there is another important component to this relationship, which is the microbiota, or the millions of microorganisms that reside within us. So what if I tell you that in order to be healthy, both physically and mentally, you need your diet and microorganisms to be in harmony. Without them, don't expect a healthy life. So now comes the question, what drives their relationship? Or as the title of my talk goes, what are the missing links between diet, health, and microbiota? And so that is what I'm trying to find out with my PhD in biochemistry at MUN. So if you look into my slide, in the nice little human figure to the left, you can see that our brain and our intestines, or the gut, are linked to each other. The brain communicates with the gut and vice versa. So different research work done in this field have shown that if there are any changes to the gut, there will be changes in our brain functions. And if there are changes in the gut or in the brain overall through this link, there will be different gut-related problems as well as brain behavior-related problems that lead to so many disorders that we have come across or we read about nowadays. So zooming in from that little figure into the intestines, the next figure, you will see that we come across two components. So we have diet, that is the food that we consume, and the microbiota. Zooming in further from that into the intestinal cells, or the cells that make up our intestines, we'll see that both the diet and the microbiota component are present. Now, we have the intestinal lumen part to the top, which is where the food passes. And then we have the blood side on the other side of the cell. So what happens here is that with the food that is coming in and with the microorganisms that are also present in this layer here, in the intestinal layer, these two components, they produce an important class of chemicals known as trace amines, which is the interesting part of my talk and which is what I focus on for my studies. So these compounds are important because they help in maintaining psychiatric, metabolic, and immune-related functions. But how do they do that? So before explaining that, I would like to bring up the important biomolecules that we have all studied during our school, which is protein. So if you remember, there are different kinds of protein present in our body. We have proteins that provide protection, proteins that are involved in maintaining structure. We also have proteins that transport molecules across the different cells of our body called as transporter proteins. We also have proteins that transmit signals throughout our bodies called as receptor proteins. So, well, if we go a little bit detail into my project, these important chemicals that I talked about, the trace amines, what they do is they activate a receptor protein called as TAR1, which is present inside the intestinal cell. Now, this receptor protein, TAR1, is important in maintaining psychiatric, metabolic, immune-related functions. But how do the trace amines coming from diet and being produced by the microbiota have access to TAR1? So that is where the missing links come. The transporter proteins that transport trace amines into the intestinal cells act as the missing links. So with my research, by using different biochemical assays and experiments, I've been able to find out that there is a transporter protein that moves trace amines into the cell from the lumen. And this has a very complicated name, but I'm just going to shorten it and refer to it as OCT2. And this transporter protein is important as this can now act as a target to maintain the different functions that I talked about: psychiatric, metabolic, and immune-related functions. Now, in addition to that, trace amines are also produced by our body itself. And that is how they can also be found in the blood. So that means in the other side of the cell. So what happens is, through my research, I've also been able to find out that there is another transporter involved in transporting trace amines from the blood and into the intestinal cell, which enables it to access the receptor protein called TAR1. So this is another missing link. However, I have not been able to identify the transporter protein yet, which is an extensive part of my project. In addition to that, I'm also trying to use different computer softwares and basically use technology to find out if there are any other transporter proteins or missing links that can transport these important chemicals into the intestinal cell. So the significance of this study for my project, or these missing links, is that once they are identified, they can be targeted by developing drugs towards them or against them to cure different classes of disorders that include anxiety, depression for psychiatric disorders, autoimmune disorders, as well as metabolic disorders like obesity and diabetes. So that is what I wanted to share with you all, and I hope this has been educative enough.
Thank you so much, Shreyasi. It was very interesting research, and thank you for your presentation. Now we can have Kris.
Well, thanks very much. So I'm not sure what each of you thinks about when you think of physics, but I wanted to tell you this afternoon about what makes physics so amazing to me. And so first of all, as you just heard, as my job, I lead a team of people who come to St. John's, to Memorial University, from all around the world just to train to become scientists. And I find physics a fascinating kind of science because I get to understand and predict things that happen in the natural world. And in my team, we work with light. And in particular, we use light with special colors to help understand how stuff is put together. Now, the light that we use isn't visible to humans. It's called infrared light. And it turns out it's the same kind of light that you have in your TV remote control. And I can even show you. So I have here a TV remote control. And when I point it at my webcam, you can see a little bit of a blinking there. And so even though we can't see infrared light with our eyes, webcams and our TVs, and it turns out a lot of other kinds of devices can see infrared light as well. And so it's good for more than just changing channels on your TV, of course. Turns out it's just the right energy for jiggling atoms. And those atoms are the building blocks of everything that's around us. So I can give you an idea of how this works with the animation that I've got here on the screen. And so you can give a thumbs up if you have seen this dance and you think it's awesome. And if you're sick of seeing it, you can give it a thumbs down. Oh, look, everyone's being positive. There's lots of thumbs up. So it's called a flossing dance, right? But in fact, this is surprisingly like the kinds of jiggling that groups of atoms can make when they're inside a solid. And this regular swinging back and forth motion is essentially what atoms are doing. And so it turns out these jiggling patterns are different in different objects, and they function like a fingerprint. And we use those fingerprints to help us figure out what atoms are inside things and also how these atoms are arranged, which I just think is super cool that I can do this with light. Now, we also work with fantastically amazing machines to help us produce and collect exactly the right kind of light to help us to do this. And to show you what a physics research lab looks like, I've got a photo here on the screen that's on the right side and it's me and a student named Ben who came to Newfoundland all the way from China to train with me to become a research scientist. And the research that we did was in Saskatoon, and it turns out it was on Treaty 6 territory, homeland of the Métis, where we did our work there. And this is a photo from this lab that we came to work in. And we did really fun and very complicated experiments that used almost all of those equipment and machines that you see around us. So you can ask me questions later about what the smoke is that's in the front. It's probably not what you think it is. So what's really fun about our physics is that it helps us to understand so many interesting things that happen outside of physics as well. So we work with archaeologists to help to identify things that they dig out of the ground. We work with people who study sea creatures to understand how hard shells grow. And we even work with people who are trying to fly drones to simulate missions that might find life on Mars. And all of these, it turns out, benefit from the way that we use light to help us fingerprint different kinds of substances. So even if physics doesn't bring something to your mind before this talk, I hope now you can see that if you're curious and you like solving puzzles, that you should probably try learning a little bit more about physics, or at least give it a try. Thanks.
Thank you so much to all of you. They were all very amazing and exciting topics. I have lots of questions in my mind, but first I want to start with a couple questions that we have as a group of organizers, very general questions to each of you. So my first question will be, what would be your top recommendation for a woman studying or working towards a STEM career? We can start with Julie. You have an idea, what would be recommending to women?
So I've been thinking about these questions a lot since you posed them to us, and I don't know that this question or the other question has clear answers, but to me at least, it's kind of a two-part answer of or related answer of create a group of or find a group of people who support you along the way and help keep your spirits up, also give constructive criticism, but in a kind and way that actually you can tell that they are there for your growth. So I think it's a lot about choosing the people who are around you and making sure you choose to not be with toxic people as much as possible. And related to that of know when to listen to people, but also know when to not listen to people. Like sometimes you'll be told you can't do something, but you know you can. Like just don't listen to yourself when it outweighs what other people around you are saying. And I know that's a hard thing to do, but it comes with practice. And just don't see everything in science that leads to failure in a lot of ways, or at least at some point will lead to failure, and just don't take it personally. Like there's failure in science and that has nothing to do with you as a person. That has to do with, the world. It's normal for failure.
Yeah, I think that was a great point. And you're also right that there is no one true answers to any of those questions. And we just want to know your experiences, your feelings and thoughts about those questions. They're very, they might be subjective, but they might be objective to other women listening to us or will listen to us in the future. So thank you for your answer. And also I want to quickly remind that the people who are listening to us participating in our conversation, they can send their either general or specific questions to each speakers through chat box. And I can ask them to our speakers. So Shreyasi, what's your opinion on this question?
Yes, so I think I have so many things to suggest to women starting their career in STEM, but the top one, what I would say is, I think that empowering men to take equal responsibility in family life also plays a big role, and it shouldn't only be the females, the woman's duty. So that could lead the woman to focus better in the career that she's starting, particularly with STEM, because you would need to be very confident, persistent, and patient throughout the process. So you cannot expect to, just take responsibility for a whole different family life, which adds a lot of pressure at the same time, in addition to the STEM work that you're doing. So I would think that being persistent, confident, and patient would help. And at the same time, if you can, empower your male member, or whichever male member in your family, to take equal responsibility in family life.
Yeah, thank you so much. And so what's your insight in this topic, Kris?
Yeah, I think it's important to talk with other people. I'll call them mentors, but it always may sound like a funny thing to define. Basically, what I found is that doing science after being an undergraduate or after being out of high school is totally different than what it's like to take classes. So talk with people who are doing science in different kinds of ways to see what their job is like, to see what they like about what they're doing, because it's so different from the classes. And as everyone said, this is much more than a few minutes to say this is the best advice. Finding people who can help you learn the ropes and provide perspective is really important all the way along, and you can get used to it by starting to work with mentors early on. And you'll also find that there's always going to be someone that you can mentor too. So even if you are in high school and you feel like you haven't started anything yet, there's probably people that you know that are much, much younger that don't even know that science is a thing. So there's always an opportunity to give back too.
Thank you so much. That was a great recommendation. So since you gave your recommendations to women considering working or already working in STEM, I just want to learn your opinions about your future selves. What would you recommend yourself 10 years from now on? Like, what would it be if you had a chance to send a letter to yourself, your future self, or have an opportunity to have a little chat with yourself? What would you recommend your future self? So, Shreyasi, do you have an idea?
Well, I would really tell myself to focus on the basics really well. Because right now at this stage, I feel like whatever I did in school, high school, is what everything matters. I mean, it's just you keep on going forward, just advancing and nurturing the basics that you have learned. But I feel if you want to stay in STEM, you need to know your basics well. So I would say like high school, college is really important for a career in STEM. And if I had an opportunity to go back, I would definitely go and, tell myself to be more focused and, try to just memorize them and not forget them ever. So that's something that I would say to myself.
That's so nice. So how about Julie?
I think I would probably tell myself to keep remembering something I'm trying to tell myself now, which is don't compare your successes to other people's successes. People have their different ways of doing things and their different value systems that make them prioritize the things that they do. And so it's not really fair to compare yourself to other people. Just make sure you're have an idea of, for yourself, what you consider success and strive towards that.
That's true. So what, Kris?
Yeah, I think I would have to say keep doing what you're doing. There's so many different ways that your life and your career can go, and they're all good. And it seems at the time like one choice is going to make or break your future, and I don't think it does. You have to be flexible. So that's the main thing I say. Would probably tell myself to keep being flexible. But if you're curious and you still have a smile on your face, you're doing a lot of things right. So
Yeah, that's so correct. Thank you so much for your opinions. So now we can go through the audience questions now. There are some specific questions to you guys. So first, to Julie, question is, what kind of threats the hyenas face? Do humans actively hunt them, or do humans just affect them by being around and in the area?
That's a great question. Naturally, like leaving humans out of the equation, the biggest mortality source to hyenas is actually lions. They're their biggest competitor. But especially in areas with humans, humans have a huge impact on hyenas. And they don't hunt them per se, but because hyenas are a threat to their livestock, they will kill them frequently on site. There's lots of like spearings that you'll see happen, and sometimes when there's been, for instance, a lot of killing of livestock, there'll be poisoning events where farmers or herders will poison a carcass, and in the attempts of getting hyenas to eat it. And of course hyenas will, but so will a lot of other animals. So that this leads to kind of mass mortality events, which aren't ideal, but at least from my perspective, I totally understand why the herders are doing what they do. If it were me, I would use a spear over poisoning, because poisoning is indiscriminate, but that's another issue. But also just being in the area affects hyenas in both positive and negative ways. So having more humans in the area leads to more trash. And hyenas they love the trash, because you don't have to hunt it, you can just eat it. We have seen so many different things in hyenas' stomachs when we've done necropsies, once they've been found dead. And it's kind of like they're like land sharks. You'll hear stories about finding like license plates in sharks' stomachs and stuff like that. And we found license plates in hyena stomachs because they have these crazy strong jaws and they just chew and eat stuff. They're crazy animals. So like they benefit from the trash, but they're also at a huge risk from having humans around them. We see that their behaviors are changing just by having humans around. They're becoming more nocturnal. Their sociality is changing. And that's just to name a few things, but they have distinct behavioral and hormonal changes when there's more humans in the area.
Yeah, that's interesting and very exciting. I also have another specific question to you and to your research. It's more like a maybe evolutionary way or philosophical way of understanding it. You mentioned that there are lots of similarities between humans and other wild animals who lives in groups. So since humans live in groups starting from the very beginning of the history to present day, what are the main behavioral similarities that we are talking about here with those so-called wild animals? So are we one of them? Like in what ways that we share those wild instincts, or in other way, are we share the emotional bond and like very strong emotional feelings as well? Like what are the main similarities that you would observe?
So there's a lot, off the top of my head, things that I think of are like, how in humans we talk about socioeconomic status, so that has to do with your social status combined with your your wealth status in many ways. And hyenas live in a hierarchical society based off rank, and their rank is so similar to the socioeconomic status. Like the way we see that those that come from a higher socioeconomic status keep staying in higher socioeconomic statuses. That's the same thing with the rank hierarchy in hyenas. If you're born to a high rank, you do better, you stay in that high rank. And those that come from a low rank have a very hard time overcoming it. And this is not just in hyenas, this is in basically any rank-based society. That's very hard to get out of the status that you're born into. But to me, humans are not that different than any other animal, because humans are just one type of animal. And I guess we're lucky in being humans in that we can talk to each other and understand each other. And so we can get deeper insight than we can observing other animals. But I think it's unfair to think that they're not having these also they have their own type of, strong connections. They are, though it may not be the same as humans, it's just as important to them in their survival as it is to us in our survival. If that kind of answers your question. It is a very broad philosophical question, but.
Yes, but yeah, you're very good at explaining it. So yeah, that's very interesting that how we live all together, like in the same similarities in our emotional world. Thank you so much. And I have another specific question from the audience to Shreyasi. So is there a specific food group that contributes more of the trace amines? Are there certain foods we should avoid because of this?
Yeah, so trace amines are really abundant in fermented food. So like we have cheese, red wine, meat. So trace amines are really fermented in fermented food groups, basically, because the way they are produced is by, not to go into real science, but by breaking down the different components that make up proteins in the food itself. So that is how they're produced. So those are the food groups which technically are abundant in these chemicals. I would not say that it would be a good idea to avoid these chemicals, because trace amines, as I mentioned, they are important in maintaining psychiatric, metabolic, and immune related functions in our body. They may help maintain all the reactions that actually control our immune related reactions taking place, or our brain behaviors. So I wouldn't recommend avoiding them, but if that answers your question, those are the food groups that are abundant in these chemicals.
Nice, thank you. And Kris, if no one is asking from our audience, I really wanna ask that question. What was that smoke in the photo?
Yeah, good. It was liquid nitrogen, and it was quite fun because the experiments we were doing, we needed to do at low temperatures. And so in fact, what we ended up doing was using most of that equipment in the room in order to direct our light on a very, very tiny sample. And all of that was the liquid nitrogen. We had to fill that up. So just watching how they fill up these large containers, if you've never seen it, it's great fun. All sorts of protective equipment, gloves, face shields, coats and everything like that. And the other thing that's interesting is that this lab where I was working it operates 24 hours a day, 7 days a week. And it's an international facility where people go to do experiments. And so when Ben and I were there, we had very short amounts of time. And so we had to work around the clock. And so sometimes we were doing things like working with people to help fill these things at 2 o'clock in the morning. And I think our very first fill did happen at about 2 o'clock in the morning. And so you're trying to stay awake and you're trying to watch all these cool things. You didn't want to close your eyes because there was always something exciting going on.
Wow, that's exciting, definitely. Yeah, like I think I'm not an expert or anything in physics, but my two favorite things, one is hydraulic press that I love to watch to be crushed by hydraulic press. The second one is liquid nitrogen. Just those two things making amazing, crazy, and fun stuff. So
well, you would have enjoyed this because we also used a hydraulic press. It turns out that the samples that we worked at to direct the light on, we did need to press them into tiny little pellets. They ended up looking like pills by the time we were done with them. But yes, it involved a hydraulic press. the reason I said it might not be what you think it is, we also did experiments in that same run where we heated our sample up very high, and we did in fact at one point smoke that was a bad kind of smoke coming out of our cell as well. So we did have all sorts of excitement on that particular round.
Oh, wonderful. So I have another question to you, Kris. Since you mentioned both in your blog and in your speech that you always loved solving puzzles and you're enjoying solving puzzles. So my question is, what was the most exciting, fascinating puzzle you solved in your research or in your career as a professor and mentor? Like, it doesn't have to be very solid puzzle kind of thing. Might be just like a relationship or like whatever comes into your mind.
Yeah, there's certainly scientific questions that have been fascinating to figure out that have taken a bunch of people to put together. So one thing I can point out is when I started working with people who did archaeology, it turns out that this kind of infrared light measurement was at the heart of that. And so I was working with a group in Israel, and it was a mixture of chemists and people who did archaeology. And I was there spending a year on sabbatical leave, and I was trying something new. It was kind of risky and new and different for me because it was very different from what I had done before. And it turns out that they had found a neat way to use infrared light that would help them basically take up something that was like a white rock, and they could use the infrared light to tell them whether it was a plaster floor or a piece of limestone. And so this is a huge implications. So if I had a floor that was a nice plaster floor that people made and walked on, that's a really different interpretation than if I just had a rock that happened to be crushed and flat. And so they knew it worked. They tested things in the labs, they tested things in the field, they knew it worked, but they didn't understand why. And so in the year I spent there, I learned an awful lot about all sorts of things, and I did help solve the puzzle by getting everybody's different ideas together. We figured out why it works, and that's a large part of what I still use that seed in my research later on. So that's a scientific kind of puzzle. The other thing I found is that when I'm helping people learn how to become scientists, each person is a puzzle too. And that's a good thing. Basically, if everyone's curious and everybody's flexible and you're trying to figure out how to do things, everybody finds how they learn in a different sort of way. And so that's also a challenge, and it's fun too, because when people see things that they really enjoy doing and they stay curious and you see these light bulbs go on over their heads, it's one of the best things ever.
Yeah, that's great. Like, it must be really nice to work on practical science. I hope I'm using that word correctly. So for example, I'm doing behavioral neuroscience, and for me to see the results immediately, it feels impossible. So I'm working in a very challenging area of human mind and brain. But like, for example, like biology, chemistry, and physics, like you can see your results either immediately, or you can see that, okay, it's not working, so I need to change my way of experimenting this. So that must be really like satisfying. And yeah, I'm envious, you guys. So good. Thank you for your answer. And I want to wrap up very quickly, but first I have another question for Shreyasi. And if our audience are not asking any more questions, I can wrap up the session for today. So Shreyasi, I have it's more like my question, not a general one. So you mentioned that there's a connection between our intestinal, digestive system in our brain, but for decades that we kind of knew that if we're under stress, if we have anxiety, it will affect our stomach, but not the other way around. And I'm assuming that this is a newly found fact. And So if also our guts affecting our brains and our brains or mood affecting our guts, and if it is just a vicious cycle, where should we start fixing our problems? Like, should we start like focusing on our mental health to not to have gut problems, or should we focusing on our gut problems to not to have mental health? What would you say?
Yeah, that's like the chicken and egg puzzle. But well, from the extensive literature review that I did as a part of my thesis work, I believe, as has been mentioned, that every disease begins in the gut. So I would say, from the research literature review that I have done for my thesis, so many research has reported that the microbial profile is so vital in so many conditions, and they're implicated in so many disorders in so many ways. I would say controlling your diet is the way how you can, achieve a good mental balance as well. And not to forget the other health aspects like your metabolic health or your immune health. But even if it is mental health, I would suggest focusing on your diet, because microbes are all, I would say, which controls every pathway in your body. So I would say going from the gut to the brain would be, starting from that direction would be ideal. So just focus on your diet, and that should create a lot of advantages.
Okay, thank you for your answer. Like, maybe it's an universal phrase, but we have a wording that you are what you eat. So
Yeah,
I
exactly.
think that's just giving the point with science. So thank you so much. We have another question from our audience to all of you guys. So what is everyone's favorite part of being a scientist, or the science field in general? So if you have an answer, you can just unmute yourself and answer the question.
I love the fact that my job is different every single day. That part is really neat.
Perfect. Yes. I can see the satisfaction on your face as well. So that's a great thing. It means that you're doing something great and you're happy with yourself. It's just the amazing outcome of science, I guess. So Shreyasi?
Yeah, so I like the fact that I learned so much by being a scientist, like not just in what I'm interested in, but other things as well, like non-science fields too, which I don't really get time to catch up on, but you get to use some aspects of everything in science. So that's what I like learning a lot.
Yeah.
I second what both of them said. Those are both on my tops. I'll also add getting to meet and work with people who are all over the world and very different from me and think different ways. I like also learning about the people I'm working with as much as I do what I'm working on, though I also study social behavior, so I feel a little biased.
So thank you, everyone. That was an amazing session, and I enjoyed all of your presentations and your answers. So again, thank you so much to three of you for sharing your time and your experience with us. And also, I want to thank to our audience as well for their amazing questions and engaging with us today. Also, I want to quickly mention that we're so thankful to Wildlife Evolutionary Ecology Lab, who donated this Zoom account in kind so we could host this event online. And also many thanks to the St. John's Farmers Market. They were willing to have us before pandemic in their area. So maybe next year we will be there if they want to collaborate with us again. And also, speaking of the future events, if you're interested in any aspects as becoming a volunteer or an organizer or a speaker for the next year, you can definitely contact with us through our Twitter account or through email. And we would love to have more people, because me, Christina, and Sarah, we enjoyed a lot to being an organizer, and also bringing this kind of an event to St. John's for the first time. And also, if you have any feedback to us about the event, about the sessions and other stuff, we would love to hear them. This was our first event, so we would like to learn from you and improve ourselves, and maybe for the next years it will be a better and a bigger event for us. So thank you so much again for joining. I hopefully see you all for the next year's St. John's Soapbox Science St. John's event. Thanks. Bye-bye.